Okay, here we have a classification system, life as we know it, and we are talking about the eukarya domain, and we are focusing on the animal kingdom. And through the different classifications, we can actually see that we have phylum, class, order, family, genus, species in order to give us that exact organism that we're talking about. In the animal kingdom, I actually want to go over the different phyla with you guys. And there's actually 36 recognized phyla for the animal kingdom. I'm going to go over the most common 9 of them. These 9 that I'm going to go over house the majority of animals. And the 9 phyla, of course, will have their own class, order, family, genus, species. This differs per animals, but the phyla is how I'm going to classify animals from here on out that are shown to the left here in this image. Phyla are classified by body plans. Body plans were used to construct phylogenetic trees, showing those evolutionary relationships among the animal groups. So what do I mean by body plans? What I'm talking about is, does that organism have tissues? Tissues are a collection of cells that perform a specific function. So think of skin tissue, think of muscle tissue. So does that organism have true tissues or not? So that's a way to classify these animals. Does that organism have radial symmetry? I'll go over what that means, or not. That's another way to classify them. Do they have bilateral symmetry? And so forth. This is a system that biologists use to classify these different phyla. This is a great mechanism that still holds true today. I am going to teach you these different body plan mechanisms that we use to group these different animals. More recently, genetic data is going to really help us determine how related these organisms are to each other and how we can modify and refine these groups, kind of make them even more perfect than they are. But these body plans were a great system because it turns out through the genetic data, scientists were pretty close at classifying these organisms. The 9 phyla I'm going to go over, as you can see in the flow of the lecture, are going to be sponges, cnidarians, mollusks, flatworms, annelids, roundworms, arthropods, echinoderms, and chordates. So let's get to it. Body plans, let's go ahead and get started. A tissue is a group of different yet similar cells that come together to perform common function. So here I'm showing you nervous tissue as an example. In nervous tissue, I have cells called neurons that are performing a function. I also have immune cells in nervous tissue that are performing a function. I have supporting cells that are located in nervous tissue. All these different types of cells are coming together to perform a common function. So in the evolutionary timeline, I have an ancestral protist, that colonial flagellated protists that I discussed during the Cambrian explosion, that's going to be the ancestor of animals. And along that timeline, I acquired tissues in order to give us 8 of the different phyla. One of the phyla does not have true tissues. It is the only one that does not have tissues. So to the right of that homologous structure are sponges. The next homologous structure I'm going to see is radial symmetry and bilateral symmetry. So we can actually group the different phyla based on that. Let's go ahead and discuss what symmetry is. Radial symmetry has to do with an organism is going to have a radiation of body segmentation from the center. So what I mean by that is that any slice, let's say I look at this creature right here and I take a slice right there in the center. The left side is going to be similar to the right side. Well, let me slice it another way. I slice it this way. The left side will be similar to the right side. So in radial symmetry, any slice through any central axis divides it into mirror images of each other. I can actually see that with a flower pot. If I slice it this way, the left side is the same as the right side. It's mirror images. If I slice it the other way, this side is going to be similar to the other side. So no matter which way you slice it, centrally, you're going to get mirror images. That's called radial symmetry. Bilateral symmetry is different from radial symmetry in that only one slice is going to divide it into mirror images, mirror halves of each other. So that's this crustacean here. If I actually cut it this way, this side, I'll go ahead and say the right side of that creature is going to be mirror images to the left side of that slice. But let's say I sliced it this way. Well, this side is quite different from this side, so I can't slice it that way. So there's only one way I can slice it to get mirror images. That's called bilateral symmetry.